Sampling is the idea that you're selecting a part of the whole, because you can never really do research on the entire population at once. You always work with kind of small bits and pieces of it. And what we do hope is that this small sample can actually represent the whole, and we can generalize our findings to everybody that we want to really study. Now, there's 3 steps to actually creating a sample. First is selecting your population. These are the people who you want to generalize your findings to. It can be very large, it can be states, provinces, countries, it can be the entire world, or it can be very specific. It can be a small group of people who come into one very specific clinic for a very specific disease. And part of this process, it takes time, it takes very concerted effort to make very good decisions in terms of what your population looks like. But part of this process is identifying your unit of analysis. We oftentimes measure individuals, but sometimes we measure groups of individuals, households, communities. Sometimes it is even on the country level. And once we get outside of this individual level, we start talking about things like ecological studies. And you just got to keep that in mind, both when you're doing your sampling as well as when you're interpreting your data. Now, the second step in creating your sample is identifying your sampling frame. Now, your sampling frame is everybody in your population who can actually be contacted. Now, your population can be very big, it can be very small, but not everyone can actually be reached. Some people just may be unreachable for whatever reason. So your sampling frame, whether it's an actual list, a physical or virtual list, or some other way to contact people, those are the people who will really be part of your study. And you hope that they represent the whole population, although you can never be 100% sure. And finally, to create your sample, once you have your sampling frame, once you've defined the population, you need to pick a sampling technique. There's 2 broad categories, probability sampling and non-probability sampling. And then within each one of these categories, there's multiple different subtypes and very specific ways to do the sampling, and we'll talk about those in other videos. Now, why do you want to do this well? Why sample at all? Why do it in a good way? Well, you do it so your study has good external validity. And external validity is the ability to generalize your findings to the population, which is really the entire point of doing research. And if you do sample well and you can generalize the findings to your population, you can often create very informative and very productive studies. But there are other threats outside the sampling procedure and part of the sampling process that you do have to concern yourself about regarding external validity. The biggest one is people, because people don't always tend to do what you want them to do. Sometimes you contact them because you sampled well and you've reached them, but they just don't want to participate. And those people who participate can be much different than the people who don't. Again, not everybody in your population is contactable, so your sampling frame could be different than the population itself. If you do get people to be part of your study, they might drop out. And the people who drop out of your study before they finish might be different than the people who complete it. Interventions don't always work the same on everybody. And then the thing that all researchers do have to worry about when they do sampling is sampling error. And this is the idea that any characteristic of a population basically fits the classic bell curve, fits the normal curve. And while most people are in the center of that curve, there are people on the edges too, on the tails. And it's entirely possible for you to do sampling well, to have a well-defined population, have a really robust sampling frame, do the sampling methodology exactly as indicated, but find some of these really random people over at the tails who don't actually represent the population itself. So you'll hear more about sampling error in a separate video. Now, place, especially when you're doing an intervention or you're looking at exposures, they don't always work the same everywhere. Clinic settings are much different than doing population-level interventions. Cities are much different than the suburbs, which are much different than rural locations. And things that work in developed countries often don't necessarily work in developing countries. And finally, time. Something that we don't always think about is that when interventions are first developed, that's under much different circumstances and environments than when they may be implemented on a wide scale farther down in the future, and particularly things like smartphone apps. Interventions that could have worked 10, 20 years ago were created before the invention of smartphone apps. And now that these apps are being created, we don't know what they're going to look like or if they'll still be effective 20, 30 years from now. Now, there are things we can do to improve external validity, and most of this has to do with just doing a really good sampling procedure. Have everything well defined, use random samples whenever possible, so use a probability sampling technique, which we'll talk about in another video. You can actually narrowly define your population of interest. This helps you to generalize your findings to them, although potentially decreases the importance of your study just because it's not applying to as many people. And maybe most importantly, because you can actually kind of control for sampling error, is to replicate. Take multiple samples, redo the studies, ask the questions again, and then ultimately you'll come down to, and the average of these results will be kind of the population parameter. And finally, you can actually test whether you do actually have good external validity. So once you've collected your data, you can see if the characteristics of your sample, whether it's demographics or behavioral characteristics, actually match the population proportions you'd expect based on available census data and available surveillance data.